Students, in this note set, we're going to talk about how a cell divides. At this point in our study, we've talked about the DNA molecule, how it's used to make messenger RNA, and then from messenger RNA to make proteins. Now we're going to take a look at how a cell copies itself prior to dividing and making an entirely new cell. The relevant reading can be found in Chapter 8 of your bio book, sections 8.1 to 8.5. First, let's ask the broad question, what is cell reproduction? Well, if we know that having cells was one of the main characteristics of being a living organism, and that cells are the functional units of all living things, then to reproduce cells is the basic process that needs to happen in order to make an entirely new organism. Cell division refers to the distribution of genetic material and cell components to the new cells, which we term daughter cells. Daughter cells are the new cells that are produced from the parent cell prior to cell division. As a reminder, the DNA molecule is a double helical molecule. It consists of two strands, which are linked together by the bonding between nitrogenous base pairs. And as you know, adenine pairs with thymine on the complementary strand, cytosine pairs with guanine on the complementary strand. A DNA molecule is typically hundreds of millions of base pairs long. So it's a really, really, really long molecule. And to help it get packaged and condensed in the cell, there are proteins called histones around which the DNA molecule wraps itself so that it can stay organized. It's a bit like a spool around which you wrap a wire, a really long wire, in order to keep the wire organized and not tangled up. Okay, so the DNA itself, a single DNA molecule consists of a double helix, but when you copy it or replicate it or duplicate it, all of those words mean the same thing, you'll end up with two double-stranded molecules. Condensing the DNA into an organized format is yet another consideration. So DNA will duplicate prior to condensing. The two copies of DNA, exact copies of each other, are called sister chromatids. It's as if you put a picture on a Xerox machine and hit print. You would get an exact replication of that picture that you put on the copy machine's platform. Not a single thing ought to be different if the copy machine is working right. Well, that's the case with DNA replication. The two sister chromatids are linked together. They're joined at an area called the centromere, which will be important as we pull the DNA to opposite ends of the daughter cells into opposite daughter cells in replication. So the two duplicate copies or replication results are sister chromatids. They are joined together at a site called the centromere. Now, the centromere doesn't have to be at the center of the DNA molecule necessarily. In fact, take a look at these high definition microscope images. Pictured here are 5 different types of DNA, different chromosome types. Notice, first of all, there are different lengths. Some DNA molecules are longer than others, even though they're all very, very, very long molecules. Secondly, notice that the centromeres are not always exactly in the middle. In fact, this chromosome to the upper right of the screen is a pretty short chromosome, and its centromere is located almost at the tip of the molecule. You don't need to know the exact specifications, although if you get into molecular biology, you would definitely want to have that at least as a reference. Okay, let's talk about division in simple organisms. The prokaryotic organisms, those without membrane-bound organelles and without a nucleus. Prokaryotic organisms do still have DNA, and the DNA does, just like in eukaryotic cells, duplicate prior to division. The genetic material simply separates into two halves of the cell, and then the cytoplasm, if you take a look at the border of this image here, the cytoplasm is pinching in right in the middle of the cell. The binary fission process by which prokaryotic organisms replicate is fairly simple and straightforward. The process by which eukaryotic organisms is a little more complex, and we're going to study that one as well. It's called mitosis. First, let's diagram binary fission. If you've got your cell and its typically circular piece of DNA attached to the cell membrane, we would send, there are DNA replication enzymes that come along and read the entire DNA molecule. So one of the two strands is read as a template. The new strand is produced. And now if you've made a new DNA strand, you've got two attachment sites. They're going to migrate apart from each other, and as they do so, the cytoplasm pinches off in the middle of the cell, eventually resulting in two entirely new cells. In binary fission, we replicate the DNA and then divide cell contents equally into daughter cells without a whole lot of worrying about whether everything's exactly equal in the cytoplasm contents. In eukaryotic cells, there's a little more to it. So let's take a look at that now. In mitosis, the process by which eukaryotic cells replicate, we're producing genetically identical daughter cells. Another word for that is cloning. We are cloning the daughter cells, or cloning the parent cell to make two daughter cells that are exactly identical. We need mitosis for a couple of reasons. One, we've got to grow. In order to go from a small baby to an adult, you need to produce more cells in your body. Your body has a certain number of trillions of cells for most of us that would increase over the course of your growth cycle. Mitosis is needed to produce that. And secondly, even once you've reached your full adult height, you're going to continue to need mitosis to replace cells that die or through natural causes are sloughed off. So your skin is replaced on average every couple of weeks. And as a result, your skin cells are some very actively dividing cells in your body in order to replace the lost cells. And that process by which they are replaced is mitosis. Here's a diagram showing a cell's life cycle. Let's say, since of course it's a circle, but let's say a cell that we're going to consider begins at the end of its parent cell cytokinesis, when its parent cell completed cell division. The first phase is a growth phase called G1. And that's when a cell it's doing its normal cellular functioning. If it's a skin cell, it's being a skin cell. If it's a bone cell, it's being a bone cell. If it's any other type of cell, it's doing the normal functioning of that cell type. Some cells, though not all, are able to go into a non-dividing or dormant phase. That would occur some point during G1 growth if that were possible for that cell. And then it would just re-enter the cell cycle and continue. Then, after the first growth phase, there's a phase during which DNA is duplicated. So you double the amount of genetic material without necessarily, as we'll see with meiosis, there's another type of cell division. The number of types of chromosomes that are unique to that cell doesn't change upon duplication. After duplication occurs, there's another growth phase called G2. And altogether, the G1, S, and G2 growth phases are called interphase. Interphase is far from a boring part of the cell's life cycle. It's the major job of a cell. It's doing the work of that particular cell type. And it's certainly upwards of 90% of a cell's life cycle. But what we're interested in in this unit is the process of cell division. That's called collectively mitosis for the nuclear events. And then there's one more phase after mitosis is complete called cytokinesis. That's not an event in the nucleus, but it does complete cell division. The four phases, really quickly, that we're going to take a tour of are prophase, metaphase, anaphase, and telophase. And they go in that order. PMAT is a way to remember that order of events. PMAT, prophase, metaphase, anaphase, and telophase. Let's take a look at some of the steps. But before we do that, when we talk about cell division in eukaryotic cells, please note the two daughter cells each receive one of the two strands of the parent cell's DNA. Remember, DNA is a double helix, and in this diagram, each strand of the double helix is color-coded blue if it came from the parent cell. Well, the two daughter cells each received one blue strand, and they make their own brand new, newly synthesized pink DNA, which is the new DNA read from the parent strand's template. This is called semi-conservative replication. Okay, let's take a look at some of the phases in mitosis. First of all, between cell divisions, we call that time period interphase. In interphase, the cell is just doing its normal job, and it's the majority of a cell's life cycle. During interphase, the nucleus stays intact, but there are some tubule containing centrioles that form. A centriole is a small cell structure found in animal cells that guides cell division. Tubules that are contained in the centrioles are like train tracks along which chromosomes move from the middle of the cell to each of the ends where the new cells are formed. In each of the following pictures, you'll see a microscope image in the lower left and an artistic rendering of the same thing in the lower right. So as you can see, in interphase, the nucleus is in its normal functioning state with a nuclear envelope around it and the DNA inside. During prophase, the first of the phases of mitosis, several things happen. Chromosomes, which by this point have been duplicated, condense. That means they're wrapping around the histone proteins and getting organized in preparation for division. Secondly, the spindle microtubules guided by the centrioles are going to form, and they're going to latch onto the chromosomes. That spindle, as it's called, is the set of microtubules or train tracks that are going to pull chromosomes apart to opposite ends of the cell. Next, metaphase. The prefix meta means middle. During metaphase, the chromosomes line up in the middle of the cell, sometimes called the equator of the cell, just like the Earth has an equator. And if you take a look at the images, the microscope image for metaphase is often the easiest to tell because you've got all of the chromosomes lined up right in the middle of the cell. Stage 3, anaphase. You can think of anaphase, starting with an A, as standing for away. The chromosomes are pulled away from each other from the middle of the cell, and now they're making their movement towards the ends of the cell by the centrioles. And then stage 4 of mitosis, called telophase, telos meaning the Greek root for end, is the last phase of mitosis. New nuclear envelopes are forming around the daughter cell's nuclei at opposite ends of the cell, and at the same time, the cytoplasm is starting to pinch off in the middle. Thus, cytokinesis has begun by telophase. Telophase is the end of mitosis, but there are still a little bit of work, a little bit of number of things to do yet with the cytoplasm. Think of all the organelles besides the nucleus that are in a eukaryotic cell. Endoplasmic reticula, mitochondria. If it's a plant cell, you've got chloroplasts, ribosomes, Golgi complexes, etc. All of those need to be divided into the daughter cells as well. and it's in cytokinesis that that happens. Even though it's not considered part of mitosis, it still is part of cell division, and it's not until cytokinesis is complete that we have two brand new daughter cells.